Hello, I'm Ron Vale, professor at University of California, San Francisco, and investigator with the Howard Hughes Medical Institute. And in the first two parts of my lecture, I talked about molecular machines and even studying these machines at single molecule level. And now I'm going to a very different scale at the level of whole genomes, trying to understand very complex structures made up of many proteins, in this case, the mitotic spindle here. Now, mitosis is a wonderful problem. It's one of the oldest problems in cell biology, and in fact dates back to biologists from the 19th century. So here is actually an image by Fleming from the 19th century, remarkably accurate depiction of the mitotic spindle, showing the chromosomes and even these fibers, which we now know are microtubules. And the problem of mitosis is also very important for medicine as well. First of all, failures in the assembly of the mitotic spindle can give rise to chromosome missegregation that results in aneuploid cells. And aneuploid cells are often a precursor for cancers and uncontrolled cell division. And in addition, there are many drugs used in cancer chemotherapy that work by targeting the mitotic spindle, as I'll describe to you at the end of this lecture. So the question that I would like to discuss with you today are, what are the molecules and the molecular interactions that are needed to build this remarkable assembly of the mitotic spindle? Here we see microtubules in green, the chromosome in blue, and the poles of the mitotic spindle marked here in orange. And what I'd like to primarily focus this lecture upon is the first step of mitosis, the building of the mitotic spindle. And here it's tubulin GFP emanating from these centrosomes. And at the onset of mitosis, what you'll see is the nuclear envelope break down, and these microtubules form into the mitotic spindle. And it's that formation of the mitotic spindle that we'd like to understand, and I will discuss in this particular lecture. And there are many questions that remain in this field, even though it's been a subject of extensive study. So first of all, there are lots of microtubules that build the spindle, and we'd like to know how those microtubules are assembled. There are specialized structures in the spindle, the centrosome found at the poles, kinetochores at chromosomes that interact with these microtubules. And we'd like to know how these structures are formed. And we'd also like to know how this characteristic football shape of the spindle is formed and organized. So there are lots of questions that we need to understand. And the approach that I'm going to share with you today is an approach done in our lab to find new proteins involved in creation of the mitotic spindle using a whole genome RNAi screen in Drosophila S2 cells. And this work was executed by a very talented group of people. The leader of this project was Goto Goshima, who has now started his own laboratory in Nagoya, and Nico Sturman, who's senior research scientist in the lab that developed a lot of the technology infrastructure to make this happen. And follow-up work after the screen was performed by Nan Zhang and a new student, Sarah Goodwin. And also another key person for this work was Roy Wollman, working in John Scully's lab. And Roy did a lot of the computational analysis that I'll share with you later. But as you'll see, it's possible to do a whole RNAi screen even with a relatively small group of individuals. So why do an RNAi screen? Well, I would submit to you that you do this to try and discover something new. And I'll even use this analogy of how early explorers went out on expeditions to find new things in the world. So this shows an early world map where much of the world was very poorly defined here. There are coasts that, large land masses that have yet to be defined. This is very analogous in many ways to 20th century biology. Even as a graduate student, I had very little idea of how big the genome was, what was in there, And at that time, which was even in the mid-1980s, you could still discover a big protein superfamily like the kinesins. And a lot has changed both in world geography and in biology. In world geography over centuries, but in biology over a short span of even a couple decades. And now the world looks more like this. In biology, the genomes, through all the sequencing effort and work by thousands of investigators, we know quite a lot. The genome has now become a finite entity, and we know a lot of the components that are present in the genome. But I do want to stress that there's still a lot of undiscovered genes out there that we need to understand. And this RNAi screen that I'll share with you is a good way of making new discoveries. So let me just tell you a little bit about RNAi. It's a technique that allows you to destroy mRNA corresponding to one gene, and as a result of the destruction of that mRNA, result eventually over several days in the depletion of that particular protein. And by this method, you then can deplete a particular protein and look at a particular outcome on the cell. So the way this works is, at least in Drosophila, is that you can first make a double-stranded RNA corresponding to a particular gene, and you'd like to use a particular piece of that gene that's unique for the gene and not found in other genes in the genome. That double-stranded RNA is then taken up by cells. Then there's a special enzyme called dicer that chops this RNA into 21 base pair fragments, which then interact with a specialized protein complex called the RISC complex that then unravels these siRNAs. It then base pairs them with an mRNA in the cell, and after base pairing results in the degradation of that mRNA. And then, as I said, over a period of days results in the depletion of the protein. So we've been using RNAi in this Drosophila S2 cell line. And we've chosen this cell line for a number of reasons. First of all, it's very easy to culture. You can even grow it at room temperature. You also get very efficient RNAi knockdown in these particular cells. And we've worked out methods that make these cells very good for microscopy, which is critical for the screen I'm going to tell you about. And then if you want, you can also follow up with genetic manipulations in flies. So the screen that we did was a visual screen. What we wanted to do is to knock down mRNA corresponding to each gene in the Drosophila genome, of which there are over 14,000 genes, and then look at the effects on the morphology of the mitotic spindle. And so we did this by imaging the spindle after RNAi of each gene. And there's a lot of information that you can get from these images. You can see the overall shape of the spindle. You can measure the length. You can see if the chromosomes are aligned properly. You can measure a very special protein that defines the poles called gamma tubulin that I'll tell you more about. And you can see if there are two poles, as there should be in a normal spindle, or if the poles are split or fragmented. So there's a tremendous amount of information that you can get from direct imaging. So we knocked down each of the over 14,000 genes in the fly genome. And in this screen, we analyzed over 4 million spindles. So let me just tell you a little bit about the technical details of how this screen was done. So first of all, you need to develop an RNAi library against the entire fly genome. And this takes a lot of careful bioinformatics to design double-stranded RNAs that are unique for particular genes, for each particular gene, so you don't get cross knockdown of other genes that knock down multiple genes, for example. So this bioinformatic effort was done by Nico Sturman in the lab, and in fact now this library is available at reasonable cost from a company called Open Biosystems. And then what we did is we treated these Drosophila cells with double-stranded RNAi corresponding to each gene. So we arrayed these S2 cells in 96 well plates, each with a unique double-stranded RNA. And then to cover the whole genome, one had to use nearly 150 such 96 well plates. And we also did double RNAi with another molecule called the anaphase promoting factor. And this is a protein that's involved in the transition from metaphase to anaphase. By knocking down this protein, more of the cells arrested in this metaphase spindle, which was the structure that we wanted to investigate. Now, after we did all this RNAi, we had to then collect all the images of these mitotic spindles. And this was done with a high-throughput microscope, where we first transferred the cells to glass-bottom 96 well plates, and then put them into this robotic microscope where you could place all these plates in this robot here that will grab and place these plates then on the microscope stage automatically. The microscope will go around and automatically focus on each well, and then it will take a number of different images, again, totally automatically around the well of these cells. So we get a tremendous amount of image data, and what we image here are the DNA, we image microtubules, we image the special protein gamma tubulin that I'll tell you more about later, and another marker called phosphohistone, which is used as a marker to know that the cell is in mitosis. So out of this, we get a tremendous amount of image data, far too much for any person to kind of manually go through. So here's where computation proved very valuable. And this was code written by Roy Woolman, who wrote MATLAB code to find these mitotic spindles automatically in these images. And after the computer found these images, we did two things with them. First of all, this computer code arrayed all these mitotic spindles into this big gallery that you see here. And then the next day, after the computer collected all these images, then Gota Goshima could wake up in the morning, come into the lab, and open up these files with all these galleries of these mitotic spindles. So this is, for example, 200 mitotic spindles found from one well of one RNAi knockdown. And you could look at these images and just visually assess whether there was any defect in the shape or architecture of these mitotic spindles. In addition to this human-based analysis, Roy also wrote code that would segment this mitotic spindle and then analyze a number of features of the spindle that a computer could analyze, such as the intensity of the stain or the length of the spindle or the number of poles. So the combination of a human looking at the data and a computer analyzing this quantitatively proved to be a very effective way of analyzing a whole genome RNAi screen. So the results of the screen is that we found that there were about 200 genes that when knocked down, the protein levels were knocked down, produced some kind of defect in the metaphase mitotic spindle. And a lot of these different phenotypes are just shown in this gallery here. So first of all, I should say that we knew going into the screen that there were 49 genes that we knew produced mitotic spindle defects in these cells. And through this blind whole genome screen, we found 45 of them, which is really quite good. In addition to those 49 known genes, of this list, many of the genes that gave spindle defects are components of multi-subunit complexes like ribosomes, spliceosomes, proteasome, RNA polymerase. Interestingly, actually, the knockdown of these different large protein complexes produced quite interesting and, in fact, unique spindle phenotypes from one another. In addition, we found about 60 knockdowns of unknown or unexpected genes that were previously not known to have any role in the mitotic spindle. And this is the list that we spent a lot of time investigating. Now, I should say that all of this data is publicly accessible. It's on a website here, And you can actually scroll through and find any gene that we analyzed in the genome and all the details of the images and phenotypes that we found for that particular gene. So what can one learn from a screen? Well, of course, what you get is a big list of genes. And you can arrange these genes, as many people do, into this pie chart where you separate them into cytoskeletal or signaling proteins. And indeed, this list is somewhat useful. It gives you kind of an overview of the most important proteins involved in spindle assembly. But I would argue that this list itself really does not, on its own, provide insight into how the mitotic spindle really works. And to do that, you have to go beyond this list of genes and do more experiments. And what you're trying to find out of the screen is some unexpected result, not just a big list of genes, but some unique finding here that can give you some new insight into how the spindle is formed. And to track down an unexpected result, as I said, you have to do a lot more work. And we then did a bunch of secondary assays. If we found a novel protein, we wanted to know where is it located in the cell, for which we had to do GFP tagging. To better understand the phenotype, instead of having a fixed image of the cell, as we did in our whole genome screen, we wanted to analyze these phenotypes by doing time-lapse imaging. And in addition, we also then custom designed a number of very specific other experiments to try and understand the phenotype using additional RNAi knockdown approaches or adding drugs or doing other kinds of localization experiments to better understand the mechanism of how these genes work. So we did find a number of interesting results from the screen, far too much for me to talk about in this short lecture. So I just would like to focus on one story that came out of the screen, and that is how you make microtubules to build the spindle. So let me just tell you how the conventional view that you'll read about in a textbook of how these microtubules form. So there are two main ideas. One is that these microtubules emanate from centrosomes here, and another is that microtubules are nucleated at the chromosomes themselves. So this first mechanism, centrosome nucleated microtubule nucleation, was really pioneered by Kirshner and Mitchison in the 1980s. And it indeed is a very important part of building the mitotic spindle. These microtubules grow out of these centrosomes, and these microtubules are growing and trying to grab hold of chromosomes. And eventually, some of these microtubules make connections with chromosomes, and these microtubules become stabilized into the mitotic spindle. And indeed, you can see this by using a special tag that just binds to the very tip of a microtubule, a special protein called EB1 that just tracks along the microtubule plus N, which is here tagged with GFP. And this is a very good mechanism for following new microtubule nucleation. So here, if we look more carefully, in this blow-up here, we can see the centrosome over here. And you can see these little comets of EB1 GFP kind of emanating out of the central area. And all of these little comets that you see growing are new microtubules that are nucleating, extending out, and trying to reach chromosomes. Now we know the main microtubule nucleator there. It's a complex that's called the gamma-tubulin ring complex, which has been studied extensively. It forms this ring-like structure here. And from this multi-subunit complex, this forms a nucleating base from which a microtubule can be nucleated and extend. This gamma-tubulin ring complex then docks on to the centrosome. At the very interior of the centrosome is a pair of centrioles. There's a lot of proteins coating the centrioles, and some of these proteins then are responsible for docking the gamma-tubulin ring complex. One of those key docking proteins is a protein called centrosomin, and then this centrosome can then nucleate this spiny array of microtubules. However, we also know that spindles will form without centrosomes, so they're not essential. For example, We know that plant cells and germ cells in animals don't have centrosomes, and yet they form mitotic spindles. And even Kojikov and Reeder have shown that you can ablate the centrosome in normal animal somatic cells, and the mitotic spindle will still form. So we know something about how that centrosome-independent mechanism occurs, and that's through nucleation of microtubules around chromatin, or DNA. This was really beautifully demonstrated by an experiment by Rebecca Heald and Eric Carcenti, where they just put in beads coated with DNA and put it into a mitotic extract from frog eggs. And these DNA-coated beads, not even normal chromosomes, would nucleate microtubules, and these microtubules would reorganize themselves into a spindle-like structure. And this is also shown in these S2 cells here. I'm showing you the centrosomes here. Here is the nucleus, and you'll notice right at nuclear envelope breakdown that the right over here, you can see chromosomes, these dark bodies, and then you can see these flashes of new microtubule growth right around these chromosomes. And then these chromatin-mediated microtubules then are then integrated into the body of the spindle. However, the question is, are these the only two mechanisms that are responsible for nucleating microtubules? And here we did this experiment that produced a surprising result that suggested there may be an additional mechanism. What you're seeing here is a spindle without any functional centrosome. So we've knocked out the centrosomes completely. And what you can see here is that there are no centrosomes in the cell, but most of the microtubule nucleation is not happening from the chromosomes, but in fact is happening throughout the body of the spindle over here, in fact including at the poles. And these new microtubule comets are kind of moving out from these pole regions out to the chromosomes. So this suggested that there may be some other kind of mechanism at play, something that we really did not completely understand, but which we got some insight into from this big whole genome RNAi screen. And that's when we looked at genes that affected the localization of gamma-tubulin, this critical nucleator of microtubule assembly. So here is normal gamma-tubulin staining shown over here, and this is superimposition with DNA and microtubules, but most of the gamma-tubulin is found at the centrosome here, but there's also gamma-tubulin that's found in the body of the spindle itself. Dimmer, but nonetheless still present. Well, from the screen, we found a known gene, centrosomin, that knocked out gamma-tubulin localization to the pole, but still preserved this gamma-tubulin localization to the body of the spindle. So this was a known gene, but then we found 4 totally unknown genes, which we call dim gamma-tubulin genes, that produced the exact opposite effect. They still allowed gamma-tubulin to localize to the spindle poles at the centrosome, but they completely knocked out the localization of gamma-tubulin to the poles, knocked out gamma-tubulin localization to the spindle. And this suggested that maybe this gamma-tubulin localization to the spindle was actually specific. It involved a specific set of docking factors, perhaps these new proteins that we found. And if this was the case, that these new proteins are involved in docking gamma-tubulin to these spindle microtubules, then we would expect that these novel genes that we found would be localized to the spindle themselves. And indeed, when we tagged these proteins with GFP, we found that these new proteins were localized to the spindle. If we knocked out the microtubules with a drug called colchicine, that localization of these DGTs went away. So these new proteins, which we call DGTs, are in the right spot in the spindle to be specific gamma-tubulin docking factors. Well, now we can ask this question. Is spindle localization of gamma-tubulin by these DGTs important? Because now we had a very specific tool. We had ways of knocking out gamma-tubulin localization at the centrosome. That we can do by eliminating centrosomin, or we can knock out gamma tubulin localization to the spindle with these new proteins. And the results here were quite dramatic. Here is a normal spindle. We're looking at GFP tubulin in green and chromosomes here in red. And you can see this is a wild-type spindle, perfectly normal. The chromosomes are aligned normally. And in this next video, we're going to look at a cell that has been depleted of one of these DGTs. It's unable to localize gamma tubulin to the spindle. And what you see here is that the spindle is very much perturbed. The chromosomes are misaligned. The spindle microtubules are very weak. And this is a very abnormal spindle indeed, and quite different from this other wild-type cell. So let me just summarize what we learned here. We learned that there are actually two different pathways for localizing the key microtubule nucleator gamma tubulin. There's one pathway that localizes gamma tubulin to the centrosome. And through this screen and work from other labs as well, we know that a whole pathway of components from the centriole through other components in the pericentriolar material that dock gamma tubulin to this pole structure. But there's a separate set of components, including these novel DGTs, that dock gamma tubulin onto the spindle. And surprisingly, the spindle localization of gamma tubulin appears to be more important than the centrosome, at least in Drosophila, because we can knock out gamma tubulin localization to the centrosome and the spindle forms just fine, chromosomes will segregate. But if we knock out gamma tubulin localization in the spindle, as I already showed you, we get these very abnormal spindles. So all of this work has led to kind of at least a hypothesis, new hypothesis of how you build the mitotic spindle. I emphasize this is a very new study. It's a new hypothesis, and we'll have to see how it translates in the next few years. But the idea is that the well-known mechanisms of the centrosome and chromatin constitute the important way of getting this new microtubule started to build the spindle. You can nucleate new microtubule growth at centrosomes or at chromosomes, and that allows you to quickly build microtubules to make a spindle. But after the spindle forms, possibly one of the more important mechanisms for maintaining the spindle is that the spindle has a way of self-propagating itself. It has a way of bringing the microtubule nucleator via these DGTs onto the spindle microtubules themselves. And this complex generates new microtubules that helps to maintain microtubules in the spindle and interaction with the chromosomes. So as I mentioned at the very beginning of this lecture, understanding the mechanism of spindle assembly also has a lot of important medical implications. Many of the drugs that we currently use to treat cancer work by interfering with mitotic spindle function. Some of these, like the Taxol, vincristine, vinblastine, vinorelbine, all work by affecting microtubules and interfering with mitotic assembly. And they're very effective anti-cancer drugs. They block cell division of tumor cells and then interfere with tumor growth. However, these agents here not only affect tubulin in dividing cells, but they affect tubulin in all cells of the body. In addition to affecting other dividing cells like bone marrow, there are also a number of neurotoxic effects of these agents because they interfere with microtubule transport in neurons as well. So maybe the future is to try to develop more strategic, specific ways of affecting the mitotic spindle to stop cell division. And there are a number of other potential targets besides the microtubules, and ones that are much more specific to dividing cells. An example of this is a special kinesin motor called kinesin, a special class of kinesin called kinesin 5. And these kinesins are only used in mitosis. They have no function in nerve cells or other cells. Their specific role is to help build a bipolar mitotic spindle. And we know from studies in many organisms that if you interfere with the function of these kinesin 5 motors, you get a very abnormal spindle indeed. You get what's called a monopolar spindle, where instead of having the microtubules coming from two poles, they're coming from a single pole. The microtubules are spreading out in this radial array. And the chromosomes, instead of aligning in the center of the spindle, they're in fact aligned as a ring, such as shown here in blue. And this ring of chromosomes simply cannot divide equally to two daughter cells. And this results in a massive failure in mitosis. Now, Cytokinetics, again, I should say, this is a company, small biotech company in South San Francisco, which I'm a co-founder, shareholder, and also on the SAB, took a strategy to develop inhibitors against this specific kinesin to try to develop a new strategy for blocking tumor growth. And indeed, through in vitro assays, developing small molecule inhibitors against this motor using biochemical tools and high throughput assays, refining these small molecules, then testing them in animal tumor models, they developed an inhibitor which has been called ispinisib, which is now in clinical trials in humans. And you can see in this tissue biopsy from a tumor from a patient after treatment with this drug, this shows a stain of the DNA. And you can see that there's this characteristic ring-like pattern of the DNA indicating that this anti-kinesin drug is working in people just as it is in vitro and in animal models. And currently, these drugs against mitotic kinesins are now in phase 2 clinical trials. They've shown activity for patients with breast cancer. And over the next couple years, we'll see how these mitotic kinesin inhibitors develop and whether they will join the armament of anti-cancer therapies. And beyond these, so in addition to these mitotic kinesin inhibitors, there are likely other candidates, new targets involving the mitotic spindle that might be next generation strategies for cancer chemotherapy. Maybe these DGTs that I described in this lecture that so profoundly affect the spindle, at least in fly cells, might be candidates to look for in human cells and in human cancers to see if they might be interesting targets for new cancer chemotherapeutics. So at least I've hopefully illustrated that the connection between the study of basic molecular machines at the level of even single molecule assays, cell biological studies such as I've shown you here with whole genome RNAi, not only provides insight into mechanism, but also gives us new routes of thinking for new kinds of medical therapy.